The overall goal of the following experiment is to quantify the replenishment and mobilization of the specific synaptic vesicle pools in central nerve terminals using live fluorescence imaging techniques. This is achieved by monitoring 2 rounds of synaptic vesicle recycling using FM dyes in the same nerve terminals to provide valid internal controls. As a second step, the readily releasable pool and the reserve pool are sequentially depleted using specific unloading protocols, which allow the quantification of relative pool sizes. Next, change either loading or unloading conditions in order to study the effect of the chosen variation on either synaptic vesicle pool replenishment or mobilization. Results show that FM dye loading of different synaptic vesicle pools is reproducible based on the quantification and analysis of the evoked dye unloading.
The main advantage of this technique over existing methods, like the fluorescent family of genetic reporters, is that this technique tracks the replenishment of pools specifically by synaptic vesicles which have undergone endocytosis. It also allows accurate quantification, since this protocol is performed twice in the same nerve terminals, providing an internal control. This method can help answer key questions in the synaptic vesicle endocytosis and exocytosis field, such as how manipulation in specific endocytosis modes can affect the replenishment of both the total recycling pool and the synaptic vesicle pools within it, such as the readily releasable pool and the reserve pool.